Welcome back to our channel. So today we are going to complete the biological classification part 2. Don't worry if you miss something, you will get the notes from the link tree. So, without further delay, let's get started. Kindly refer this page for the syllabus and previous year questions topics. Introduction to Kingdom Protista. Protista includes all single-celled eukaryotic organisms. They do not fit clearly into plant, animal, or fungal kingdoms, hence they are grouped separately. The boundaries of this kingdom are not well defined. Habitat of protists. Mostly aquatic, freshwater or marine. Can be free-living or parasitic. Cell features of protists. They are eukaryotic. They have a true nucleus. Membrane-bound organelles, for example, mitochondria, endoplasmic reticulum. Some have cilia or flagella for movement. Nutrition of protists. Autotrophic, photosynthetic, like algae. Heterotrophic, like protozoans, ingest food or absorb nutrients. Some show mixotrophic nutrition, for example, Euglena is both autotrophic and heterotrophic. Reproduction of protists. Asexual, by binary fission, and sexual, by cell fusion and zygote formation. Major groups of protists. This is a short introduction for the groups of protists. 1. Chrysophytes. Examples are diatoms and dimids. The key features of chrysophytes are that they have a cell wall of silica and are photosynthetic. 2. Dinoflagellates. Examples are Goniolax, Noctiluca. The key features of dinoflagellates are that they have 2 flagella, are bioluminescent, and cause red tides. 3. Euglenoids. An example is Euglena. The key features of euglenoids are that they have no cell wall, have a pellicle, mixotrophs. 4. Slime molds. An example is Physarum. The key features of slime molds are that they are in the plasmodium stage and spore-forming. 5. Protozoans. Examples are amoeba and paramecium. The key features of protozoans are that they are animal-like, heterotrophic, and motile. Key features of Protista with their description. 1. Cell type. They are eukaryotic, which contains a true nucleus and membrane-bound organelles. 2. Cellularity. It is mostly unicellular, some colonial or filamentous forms exist. 3. Habitat. It is primarily aquatic. They are found in freshwater and marine environments. 4. Mode of nutrition is diverse, are autotrophic, photosynthetic, heterotrophic, ingesting or absorbing, or mixotrophic, example, Euglena. 5. Movement of protists. They may use cilia, flagella, or pseudopodia for locomotion, depending on group. 6. Reproduction. If asexual, binary fission, and if sexual, cell fusion and zygote formation. 7. Cell walls are present in some, example, diatoms with silica wall, absent in others, example, euglenoids. 8. Diversity of protists includes plant-like, algae, animal-like, protozoans, and fungus-like organisms, slime molds. 9. Groups included are chrysophytes, dinoflagellates, euglenoids, slime molds, protozoans. Why is Protista important? 1. Evolutionary bridge. It links unicellular organisms to plants, animals, and fungi in evolution. 2. Ecological role includes key primary producers, example, phytoplankton, diatoms, in aquatic ecosystems. Maintains food chains and nutrient cycles. 3. Medical relevance is some protists cause human diseases, example, plasmodium causes malaria. 4. Environmental indicators are that protists help monitor water quality and pollution levels. 5. Industrial applications are that diatomaceous earth, from diatoms, is used in filtration, polishing, and insulation. Let's start with the first group of Protista. First. Introduction to chrysophytes. Definition of chrysophytes: they are microscopic, mostly photosynthetic protists, including diatoms and dimids, golden algae. They are typically found in freshwater and marine habitats. Key characteristics of chrysophytes. 1. They are photosynthetic, most perform photosynthesis. 2. They float passively, they drift in water currents, planktonic. 3. Cell wall structure: diatoms have two overlapping shells, like a soapbox. Their cell walls are impregnated with silica, so they are indestructible. Diatomaceous earth is formed from accumulated silica cell walls over millions of years. Uses of diatomaceous earth is polishing, filtering oils/syrups, toothpaste, etc. Importance of chrysophytes: primary producers in the ocean base of the aquatic food chain. Contribute significantly to oxygen production and carbon cycling. Key features of chrysophytes with their description. 1. Habitat of chrysophytes. They are found in freshwater and marine environments. 2. Microscopic and planktonic means they are very small, float passively in water currents. 3. Photosynthetic means they are mostly autotrophs that perform photosynthesis using chlorophyll. 4. Cell wall structure. Diatoms have two overlapping silica shells, like a soapbox. 5. Silica impregnation. The silica makes the walls indestructible, forming diatomaceous earth. 6. Reproduction in chrysophytes. That they mostly reproduce asexually by binary fission. Why is chrysophytes important? 1. Primary producers in aquatic ecosystems form the base of food chains. 2. Major oxygen contributors through photosynthesis in water bodies. 3. Their fossilized shells create diatomaceous earth, useful in filtration, polishing, and abrasives. 4. Serve as ecological indicators, helping assess water quality and pollution levels. Second, introduction to dinoflagellates. Habitat: they are mostly marine, free-floating, planktonic. Nutrition of dinoflagellates are mostly photosynthetic, autotrophs. They have few pigments such as yellow, green, brown, blue or red, based on dominant pigments. They have cell wall that is composed of stiff cellulose plates. Flagella: they have two flagella, one longitudinal, one transverse in furrow. The special feature of dinoflagellates. Some red pigmented species, like Gonialax, cause red tides due to a sudden population explosion. Release of toxins that are harmful to marine life. Example, Gonialax. Important: red tides are indicators of water pollution and environmental imbalance. Key features of dinoflagellates with their description. Habitat: they are found mostly in marine water and are free-floating, planktonic. Cell covering of cellulose plate that is rigid. For locomotion they have two flagella, one longitudinal, one transverse. Nutrition type in dinoflagellates is mostly photosynthetic, some heterotrophic. Unique features in dinoflagellates are red tides, toxin production. Pigments of yellow, green, brown, blue or red based on dominant pigments as said before. Why is dinoflagellates important? From the base of marine food chains, red tides act as ecological indicators, toxins impact marine biodiversity. Third, introduction to euglenoid. Habitat of euglenoids are mostly freshwater, stagnant ponds. The cell wall is absent. Outer covering, pellicle, a protein-rich flexible layer. It has flagella, two where one short and another one long. Nutrition: in light it is photosynthetic. In dark it is heterotrophic, ingests other organisms. Pigment similarity: they are identical pigments to those in higher plants. Example, Euglena. Important: shows dual mode of nutrition, mixotrophic, key evolutionary link between plants and animals. Key features of euglenoid with their description. Habitat: they are found mostly in fresh water, stagnant ponds. Cell covering is made up of pellicle, flexible, protein-rich. For locomotion they have two flagella, one short and one long. Nutrition type is mixotrophic, means both auto and heterotrophic. Unique features of euglenoids is they do not have cell wall, plant-like pigments. Pigments as said before they are identical to those in higher plants. Why is euglenoids important? 1. Evolutionary link between plants and animals. 2. Capable of photosynthesis and predation. 3. Survive in varying light conditions. 4th, introduction to slime molds. Habitat of slime molds. Moist, decaying organic matter, example, rotting logs. Nutrition, they are saprophytic, feed on dead organic matter. Structure of slime molds. The body forms a creeping mass called plasmodium, multinucleated, wall-less. Under stress, they form fruiting bodies with spores. Spores have true walls. Extremely resistant and airborne. Example, not specific, general category. Important, shows features of both fungi and protozoa, acting as evolutionary links. Key features of slime molds with their description. The habitat of slime molds is most decaying matter. The cell covering they have is not permanent, but a creepy mass called a plasmodium, which is multinucleated and cell wall-less, as said before. Their locomotion is pseudoplasmodium movement. Their nutrition type is saprophytic. The unique feature of slime molds that they have fruiting bodies and stress-resistant spores. They are important because they show features of both fungi and protists. Why is slime molds important? 1. It is important because it is useful in decomposition and nutrient cycling. 2. They are model organisms in cell biology, plasmodial structure. 5th, introduction to protozoans, animal-like protists. 1. All are heterotrophs, either predators or parasites. 2. Considered primitive animal relatives. There are 4 types of protozoans. A. Amoeboid protozoans. Habitat of amoeboid protozoans is freshwater, marine, or moist soil. Movement of amoeboid protozoans is pseudopodia, false feet. Shells of amoeboid protozoans is marine forms that have silica shells. Example, amoeba, entamoeba, parasitic. Why is amoeboid protozoans important? 1. They play a role in soil and aquatic ecosystems as a micropredator. 2. Entamoeba affects human health. Key features of amoeboid protozoans with their description. Habitat is freshwater, marine and soil. Cell covering has no fixed structure. For locomotion they have pseudopodia, false feet. Nutrition type of amoeboid protozoans is heterotrophic, phagocytosis. Unique feature is some have silica shell. B. Flagellated protozoans. They do movement using flagella. Habit of flagellated protozoans is free-living or parasitic. They cause disease like sleeping sickness. Example, trypanosoma. Why is flagellated protozoans important? Cause serious diseases like sleeping sickness, trypanosoma. Important in parasite host studies. Refer the diagram down there. They have mitochondria, they need it for energy to carry out life functions. Nucleus, they need it for regulating their gene expression and cell cycle through a central control center. Kinetoplast for survival in changing environments. Flagella swim through liquid environments for motility. Key features of flagellated protozoans with their description. Habitat is free-living or parasitic. Cell covering is flexible membrane. For locomotion they have flagella. Nutrition type they are heterotrophic, parasitic. Unique feature is they cause sleeping sickness. C. Ciliated protozoans. For movement they have thousands of cilia. For feeding they use cilia to steer food into gullet. Example, paramecium. Why is ciliated protozoans important? maintain microbial balance in aquatic ecosystems, and they show advanced cellular coordination. Key features of ciliated protozoans with their description. Habitat is aquatic, fresh/marine. Cell covering is made up of pellicle and oral groove. For locomotion they have cilia. Nutrition type is heterotrophic, they ingest via gullet. Unique feature is they have coordinated ciliary feeding. D. Sporozoans. Special feature is they have no movement structures. Life cycle of sporozoans includes an infectious spore-like stage. Example, Plasmodium that causes malaria. Why is sporozoans important? They are medically significant because they cause malaria and other protozoal diseases. Important for understanding life cycles of pathogens. Important. Responsible for major diseases and form part of the parasitic food web. Key features of sporozoans with their description. Habitat they live in the host body, parasitic. Cell, covering they have no locomotion organelles. Locomotion is passive. Nutrition type is heterotrophic, parasites. Unique feature is they have infectious spore-like stage. Introduction to Kingdom Fungi. General characteristics. They are heterotrophic eukaryotes because they cannot photosynthesize and depend on organic matter. Habitat they are found in air, soil, water, decaying matter, plants, and animals. Preference they grow well in warm and humid conditions, example, moist bread, rotting fruits. Common examples, yeast, bread mold, mushrooms, Puccinia, wheat rust, penicillium. Structure. They are unicellular in yeast, exception, but multicellular/filamentous they are made of thread-like structures called hyphae. Mycelium they have a network of hyphae. Cenocytic hyphae is continuous cytoplasm with many nuclei but no septa. Septate hyphae are hyphae with crosswalls with septa. Cell wall composition they are made of chitin and polysaccharides. Mode of nutrition is saprophytic they absorb nutrients from dead organic matter. Parasitic they feed on living hosts, plants/animals. Symbiotic. 1. Lichens, with algae, mutual benefit. 2. Mycorrhiza, with roots of higher plants. Basis of classification. Fungi are classified into different classes based on 1. Type of mycelium. 2. Mode of spore formation. 3. Type of fruiting body. Reproduction in Kingdom Fungi. A. Vegetative reproduction that is by fragmentation, budding, fission. B. Asexual reproduction that is by spores are Conidia, sporangiospores, zoospores. C. Sexual reproduction involves 3 key steps they are 1. Plasmogamy that is fusion of cytoplasm. 2. Karyogamy that is fusion of nuclei. 3. Meiosis that is formation of haploid spores. Spores formed are oospores, ascospores, basidiospores. They occurs in specialized structures called fruiting bodies. Special reproductive feature. Dikaryon, N+N, in fungi like Ascomycetes and Basidiomycetes, 2 haploid nuclei stay separate in the same cell before fusing. Dikaryophase. The phase where cells are dikaryotic, unique to fungi. Why Kingdom Fungi is important. In food industry it is used as yeast in bread/beer production. In medicine they are source of antibiotics like penicillin. In environment they play the major role of decomposers and recyclers. In agriculture. Some are pathogens, example, Puccinia in wheat. Symbiosis. They play key ecological roles in mutualistic associations, lichens, mycorrhiza. Key features of Kingdom Fungi with their description. Cell type they are eukaryotic, have well-defined nucleus and membrane-bound organelles. Cell wall composition is they are made up of chitin and polysaccharides. Body structure they have are mainly filamentous except in unicellular yeasts, made of hyphae. Mycelium that is network of hyphae, they can be separated or cenocytic. Mode of nutrition. They are heterotrophic that is saprophytic, parasitic, or symbiotic. Reproduction. Vegetative by fragmentation and budding. Asexual by spores and sexual. Spore types are asexual. Conidia, sporangiospores, zoospores. Sexual, oospores, ascospores, basidiospores. Habitat. They are terrestrial, they prefer warm and humid environments. Ecological role they play as decomposers, symbionts, example. Lichens, mycorrhiza, and pathogens. There are 4 classes of kingdom fungi they are. 1. Phycomycetes. 2. Ascomycetes. 3. Basidiomycetes. 4. Deuteromycetes. Let's start from phycomycetes. A. Phycomycetes. Habitat. They are found in aquatic environments. Decaying wood in moist and damp places. Obligate parasites on plants, example, albugo on mustard. Mycelium structure. 1. Aseptate, no cross walls. 2. Cenocytic, continuous cytoplasm with many nuclei. Asexual reproduction. Spores are zoospores, motile, flagellated. Aplanospores, non-motile. Spores are endogenously produced inside the sporangium. Sexual reproduction. By fusion of 2 gametes. 1. Isogamous means that gametes are similar. 2. Anisogamous or oogamous means gametes are dissimilar. As a result, a zygospore is formed. Examples: mucor, rhizopus, bread mold, albugo, parasite on mustard plants. Key features of phycomycetes with their description. Habitat. Found in aquatic environments, damp areas, or as plant parasites. Mycelium. Aseptate, no crosswalls, cenocytic, multinucleate continuous cytoplasm. Asexual reproduction. By zoospores, motile, or aplanospores, non-motile, in sporangia. Sexual reproduction. Fusion of gametes, isogamous, anisogamous, or oogamous, to form zygospore. Examples: rhizopus, bread mold, mucor, albugo, parasite on mustard. Special feature. Spores are endogenously produced, inside sporangium. Why phycomycetes are important. Some species cause plant diseases, example, albugo. Rhizopus and mucor are common decomposers. Study of their life cycle helps understand fungal reproduction, especially spore formation. Rhizopus is used in industrial fermentation for organic acid production. B. Ascomycetes, also called a sac fungi. Common name: sac fungi. Habitat and nature. They are mostly multicellular, example, penicillium, rarely unicellular, example, yeast/Saccharomyces. Found as saprotrophic. Means they feed on dead organic matter. Decomposers. Parasites. Coprophilous, grows on dung. Mycelium. Branched and septate, divided by cross walls. Reproduction in Ascomycetes. Asexual reproduction. Produces conidia, non-motile spores. Conidia are formed exogenously on special hyphae called conidiophores. On germination, conidia give rise to new mycelium. Sexual reproduction. Produces ascospores, sexual spores. Ascospores are formed endogenously inside a sac-like structure called an ascus, plural, asci. Asci are arranged in different types of fruiting bodies called ascocarps. Examples and importance. Neurospora. Used in genetic and biochemical research, model organism. Morels and truffles. Edible, gourmet delicacies. Aspergillus, claviceps. Economically and medically important. Key features of Ascomycetes with their description. Common name is sac fungi. Cellularity. They are mostly multicellular, example, penicillium. Rarely unicellular, example, yeast. Habitat. Saprophytic, decomposer, parasitic, coprophilous, grows on dung. Mycelium. Is branched and septate, has cross walls. Asexual spores. Conidia are produced exogenously on conidiophores. Sexual spores. Ascospores are produced endogenously in a sac-like ascus. Fruiting body. Asci are arranged in structures called ascocarps. Cell wall composition. They are made up of chitin and polysaccharides. Examples: Aspergillus, claviceps, Neurospora, morels, truffles, yeast. Economic importance. Is some being edible, morels, truffles, others are used in antibiotics or genetic studies. Basidiomycetes, also called as club fungi. Common name is club fungi. They include mushrooms, bracket fungi, puffballs. Habitat. They are found in soil, on logs, tree stumps, or as parasites in living plants, example, rusts, smuts. Mycelium. Is branched and septate. Asexual reproduction. Asexual spores are usually absent. Vegetative reproduction by fragmentation is common. Sexual reproduction. No distinct sex organs. Plasmogamy occurs by a fusion of somatic cells of different genotypes. As a result in a dikaryotic, N+N, stage. Leads to the formation of a basidium, club-shaped structure. Spore formation. Karyogamy and meiosis occur in the basidium. Produces 4 basidiospores. Basidiospores are formed exogenously on basidia. Fruiting body. Basidia are arranged in basidiocarps. Examples are agaricus, common mushroom. Eustalago, causes smut disease in crops. Puccinia, rust fungus in wheat. Key features of Basidiomycetes, club fungi, with their description. Common name are club fungi. They include mushrooms, bracket fungi, puffballs. Habitat. It is found in soil, on logs, tree stumps, and as parasites in living plants, examples, rusts, smuts. Mycelium type. They are branched and septate, divided by cross walls. Asexual reproduction. Generally absent, if present, occurs through vegetative fragmentation. Absent, sexual reproduction involves fusion of somatic, vegetative, cells of different strains. Plasmogamy. Fusion of 2 vegetative cells, different strains, results in a dikaryotic, N+N, stage. Dikaryotic stage. Prominent feature that leads to formation of basidium, where further sexual processes occur. Karyogamy. Fusion of nuclei inside the basidium. Meiosis occurs after karyogamy, forming 4 haploid basidiospores. Spore type. Basidiospores, produced exogenously on the basidium. Fruiting body. Basidiocarps, specialized structures that house the basidia. Cell wall composition. They are made of chitin and polysaccharides. Examples: agaricus, mushroom, eustalago, smut, puccinia, rust. D. Deuteromycetes, imperfect fungi. Common name: imperfect fungi. Because only asexual or vegetative phases are known. Sexual reproduction. It's absent. Called imperfect as no sexual stage has been observed. Reclassification upon discovery of sexual stage. If the sexual stage is found, species are moved to Ascomycetes or Basidiomycetes. Sometimes a fungus has 2 names, one for the asexual stage, one for the sexual stage. Asexual reproduction. By conidia, asexual spores. habitat and nutrition. Can be saprotrophic, feeding on dead matter, parasites, or decomposers. Play an important role in mineral cycling by breaking down litter. Ecological and economic importance. Used in biocontrol, e.g. trichoderma. Some cause plant diseases, example, Alternaria, Colletotricum. Examples. Alternaria. Colletotricum. Trichoderma. Key features of deuteromycetes, imperfect fungi, with their description. Common name is imperfect fungi. Sexual reproduction is absent or not yet observed. Only asexual or vegetative stages are known. Asexual reproduction occurs through conidia, asexual spores. Mycelium. Septate, cross-walled, and branched. Reclassification. Once the sexual stage is discovered, they are reclassified into ascomycetes or basidiomycetes. Nutrition mode. They can be saprophic, parasitic, or decomposers. Ecological role. Many acts as litter decomposers, aiding in mineral cycling in ecosystems. Economic importance. Some are useful in biocontrol, example, trichoderma. Some cause plant diseases, example, Alternaria, Colletotricum. Examples. Alternaria, Colletotricum, trichoderma. Introduction to Kingdom Plantae. Includes eukaryotic, chlorophyll-containing organisms, plants. Most are autotrophic. A few are heterotrophic, example, insectivorous plants. Such as bladderwort, Venus flytrap. Parasitic plants such as Cuscuta. Plant cells have cell wall, mainly cellulose. Have chloroplasts for photosynthesis. Groups included. 1. Algae. 2. Bryophytes. 3. Pteridophytes. 4. Gymnosperms. 5. Angiosperms. Life cycle shows alternation of generations. 1. Sporophytic phase, diploid, 2n. 2. Gametophytic phase, haploid, n. Varies in dominance and independence. Introduction to Kingdom Animalia. Includes multicellular, eukaryotic heterotrophs. No cell wall, unlike plants/fungi. Nutrition is holozoic, ingestion, internal digestion. Energy stored as glycogen or fat. Growth results in a definite shape and size. Possess sensory and neuromotor systems, in higher forms. Most are modal, capable of movement. Sexual reproduction with copulation of male and female. Embryological development. Phyla details covered in Chapter 4. Key feature of Kingdom Plantae with their description. Eukaryotic and chlorophyllous. Contain true nuclei and chloroplasts for photosynthesis. Mostly autotrophic. Make their own food using sunlight. Some are heterotrophic, example, Cuscuta, Venus flytrap. Cell wall. Composed mainly of cellulose, providing structural support. Kingdom Plantae includes algae, bryophytes, pteridophytes, gymnosperms, angiosperms. Life cycle of Kingdom Plantae. They shows alternation of generations. Haploid, N, gametophyte and diploid, 2N, sporophyte phases alternate. Key features of Kingdom Animalia with their description. Eukaryotic and multicellular. They have complex cell structure, multiple cells form tissues and organs. Heterotrophic. They obtain energy by ingesting organic material. No cell wall. Their cells are flexible, that allows movement and specialized cell shapes. Holozoic nutrition. That involves ingestion, digestion, and absorption inside a body cavity. Neuromotor coordination. In most of the animals have sensory systems and movement ability. Sexual reproduction. Involves copulation and embryonic development. Why Kingdom Plantae is important. 1. Provides oxygen through photosynthesis. 2. Foundation of food chains as primary producers. 3. They are source of medicine, shelter, fuel, and textiles such as cotton, flax. 4. For understanding alternation of generations is key in plant reproduction and plant evolution. Why Kingdom Animalia is important. 1. Crucial for ecological balance, consumers in the food chain. 2. Provides food, labor, animals, and companionship to humans. 3. Used in medical research and genetics, example, Drosophila. 4. Understanding Animalia helps in studying anatomy, physiology, and biodiversity. Viruses. They are acellular. Not considered truly living. Because they lack cellular structure. Inert outside host. Crystalline and inactive outside a living host. Obligate parasites. They can only replicate inside living cells. They have genetic material. It is either DNA or RNA but never both. Plant viruses are mostly ssRNA. Animal viruses are ssRNA, dsRNA, or dsDNA. Bacteriophages are mostly dsDNA. Structure of viruses is capsid. They have protein coat made of capsomeres which protects their genetic material. Symmetry. They are either helical or polyhedral in shape. Discoveries are by Ivanovsky, 1892. He discovered the virus causing tobacco mosaic. Bajorink, 1898, he coined the term virus as contagium vivum fluidum means contagious living fluid. W. M. Stanley, 1935. He crystallized virus, proving its non-living nature outside host. Diseases caused in humans such as AIDS, flu, smallpox, herpes, mumps. In plants such as mosaic disease, leaf curling, dwarfing, stunted growth. Viroids. Discovered by T.O. Diener in 1971. Structure of viroids, free circular single-stranded RNA, they have no protein coat. Smallest infectious agent. It causes potato spindle tuber disease. It lacks a capsid, unlike viruses. Prion. They are infectious protein particles, they have no nucleic acid. Abnormally folded proteins that induce misfolding in normal proteins. Size similar to viruses. Diseases caused such as BSE, mad cow disease, in cattle. CJD, Creutzfeldt-Jakob disease, in humans. Key feature. They causes degenerative neurological disorders. Lichens. Symbiotic association between alga, phycobiont, is an autotrophic, photosynthetic, and fungus, mycobiont, is heterotrophic, absorbs water, minerals. Mutual benefit. 1. Algae provide food. 2. Fungi provide protection and support. Indicator species. Highly sensitive to pollution. Do not grow in polluted areas, bioindicators of air quality. Ecological role. Soil formation, pioneer species on rocks. Used in dyes, medicines, perfumes. Key features of viruses, viroids, prion, and lichens with their description. 1. Viruses. Viruses are acellular, they have nucleoprotein, they are obligate parasite. They contain either DNA or RNA, they are inert outside host, they have protein coat called capsid. They infect plants, animals, and bacteria, bacteriophages. Each type has a specific host. Viral diseases include AIDS, flu, mosaic disease, etc. Viroids. Viroids are smallest infectious agent. It consists of low molecular weight naked circular RNA, they have no protein coat. Discovered by T.O. Diener. They cause plant diseases like potato spindle tuber disease. Prion. Prion are infectious proteins. They neither have DNA nor RNA, they are abnormally folded proteins. It cause neurodegenerative diseases. They are very resistant to sterilization. They cause mad cow disease, BSE, CJD in humans. Lichens. Symbiotic association of algae and fungi. Alga, photosynthetic, and fungus, absorbs water and minerals, called phycobiont and mycobiont. They are pollution-sensitive organisms. Useful as bioindicators of air quality, found on rocks, soil, and tree bark. Why viruses, viroids, prion, and lichens is important. 1. Viruses. Causes diseases such as AIDS, flu, hepatitis. Viruses are used in genetic engineering and biotechnology. Viroids are important in understanding RNA-based infections in plants. Prion help study protein misfolding and related diseases, significant in neuroscience. Lichens are indicators of air pollution. They help in soil formation, used in dyes, perfumes, and medicine. Summary. 1. Early classification systems. Aristotle was the first to classify organisms based on simple morphological characters. Linnaeus divided living organisms into two kingdoms. First, Plantae. Second, Animalia. 2. The five-kingdom classification was proposed by Whittaker, 1969. Five kingdoms are: 1. Monera. 2. Protista. 3. Fungi. 4. Plantae. 5. Animalia. Whittaker did classification based on A. Cell structure. Prokaryotic or eukaryotic. Body organization. Unicellular or multicellular. Mode of nutrition. Autotroph or heterotroph. Reproduction. Asexual or sexual. And phylogenetic relationships. 3. Kingdom Monera. Includes bacteria, prokaryotes. They are unicellular, they have no membrane-bound organelles. Cell wall present, peptidoglycan. Nutrition, autotrophic chemosynthetic, photosynthetic or heterotrophic. Extremely metabolically diverse. Reproduce by binary fission, sometimes form spores. Some bacteria fix nitrogen, others cause diseases. 4. Kingdom Protista. All unicellular eukaryotes. Examples: chrysophytes, dinoflagellates, euglenoids, slime molds, protozoans. They have a true nucleus and membrane-bound organelles. Live mostly in aquatic habitats. Reproduce asexually and sexually. Act as a link between prokaryotes and multicellular eukaryotes. Five. Kingdom Fungi. Heterotrophic eukaryotic organisms. Nutrition, saprophytic, parasitic, or symbiotic. They are multicellular, except yeast. Body is made of hyphae which forms mycelium. Cell wall is made of chitin. Reproduction. Vegetative. Budding, fragmentation. Asexual. By spores, conidia, sporangiospores. Sexual. By zygospores, ascospores, basidiospores. Classified into A. Phycomycetes. B. Ascomycetes. C. Basidiomycetes. D. Deuteromycetes. Roles. Decomposition, antibiotics, example, penicillium, diseases, and fermentation. Six. Kingdom Plantae. They are eukaryotic, multicellular, chlorophyll-containing autotrophs. Perform photosynthesis. Have cell wall made of cellulose. Includes algae, bryophytes, pteridophytes, gymnosperms, angiosperms. Reproduction. Asexual and sexual. Alternation of generations. Gametophyte, haploid, sporophyte, diploid. Some are heterotrophic. Example, Cuscuta, parasitic, Venus flytrap, insectivorous. 7. Kingdom Animalia. They are multicellular, eukaryotic, heterotrophic. They have no cell wall. Mode of nutrition, holozoic, ingestion. Most show locomotion and nervous coordination. They store food as glycogen or fat. Reproduction. Sexual, involves copulation and embryonic development. 8. Viruses, viroids, prion, and lichens are not included in Whitaker's 5 kingdoms. Viruses. They are acellular, non-living outside the host. Living inside the host, replicating using the host machinery. Genetic material such as RNA or DNA but not both. Protein coat. Capsid. Examples. HIV, AIDS, influenza, bacteriophages. Viroids. Discovered by T.O. Diener, 1971. Smaller than viruses, consist of only RNA, no protein coat. Cause plant diseases, example, potato spindle tuber disease. Prion. They are infectious proteins, have no nucleic acid. Cause neurodegenerative diseases, example, mad cow disease, CJD. Lichens. Symbiotic association between alga, phycobiont, photosynthesis. Fungus, mycobiont, absorbs water and minerals. They are pollution indicators. Thanks for watching. If you found this video helpful, don't forget to like, share, and subscribe to support our content. This chapter is indeed vast, but don't worry, we've got you covered. We'll be posting easy-to-remember mnemonics, examples, and full forms, perfect for beginners and quick revision. Stay connected with us on social media to get updates on upcoming videos, practice questions, downloadable notes, and PDFs. Missed anything? You can access all notes and resources via our Linktree, linked in the description and our bio. Stay tuned and keep learning. Hidden Wisdom Academy Biology is here to guide you through chapter step by step.